Welcome to medical biochemistry. Proteins are large molecules made from 20 standard amino acids. From the simplest of bacteria to human beings, the proteins of every organism are constructed from the same set of amino acids. So in this section, we begin with a description of amino acids. Here shows the common structure of amino acids. All the 20 common amino acids are alpha amino acids. This is the alpha carbon. Except for glycine, the alpha carbon of each amino acid is bounded to 4 different groups: a carboxyl group, an amino group, an R group or a side chain, or a hydrogen atom. In glycine, the R group is another hydrogen atom. The alpha carbon atom is thus an asymmetric carbon or chiral center. All amino acids in proteins are L stereoisomers except glycine. The structures of 20 amino acids are shown in this figure. Please notice that these amino acids have been assigned 3-letter abbreviations and 1-letter symbols, which are used as shorthand to indicate the composition and the sequence of amino acids in proteins. We can see that amino acids differ from each other in their R groups. These R groups vary in structure, size, and electric charge, so they influence the solubility of the amino acids in water. According to the properties of their R groups, amino acids can be classified into 5 main classes. The first group is nonpolar aliphatic amino acids. This class of amino acids includes glycine, alanine, valine, leucine, isoleucine, methionine, and proline. Their R groups are nonpolar and hydrophobic. The side chains of alanine, valine, leucine, and isoleucine tend to form hydrophobic interactions to stabilize protein structure. Methionine is the sulfur-containing amino acids, and proline is an amino acid. Its secondary amino group inside the chain form a rigid cyclic structure that reduces the structure flexibility of polypeptide regions containing proline. The second group is polar uncharged amino acids. This class of amino acids includes serine, threonine, cysteine, asparagine, and glutamine. They are more soluble in water or more hydrophilic than those of the nonpolar amino acids, because the R groups of these amino acids contain hydroxyl groups or amine groups, which may form hydrogen bonds with water, but the cysteine is quite modest because it's a sulfhydryl group. 2 cysteines can be joined by a disulfide bond to form cysteine. Disulfide bonds between cysteines play a special role in stabilizing structure of many proteins. For example, bovine insulin is composed of an A polypeptide chain and a B chain, which are stabilized by 2 disulfide bonds between 2 chains and an in-chain disulfide bond. If the disulfide bonds are broken, insulin will lose its natural conformation and thus lose its activity. The third group is aromatic amino acids. This class of amino acids includes phenylalanine, tyrosine, and tryptophan, whose aromatic side chains are relatively nonpolar. Tyrosine and tryptophan are more polar than phenylalanine because of the tyrosine hydroxy group and the nitrogen of the tryptophan indurine. All can contribute to the hydrophobic effect to stabilize the protein structures. Tryptophan and tyrosine can absorb ultraviolet light because of their conjugated double bonds. The maximum absorption for both amino acids occurs near 280 nanometer. Since most proteins contain tryptophan and tyrosine, the UV absorption at the wavelengths of 280 nanometer can be used to estimate the concentration of the protein solution. Light absorption by phenylalanine generally contributes little to the spectroscopic properties of proteins. The fourth group is negatively charged amino acids. This class of amino acids includes aspartate and glutamate, each of which has a second carboxyl groups. Their R groups can yield protein and function as weak acid. Therefore, they have significant negative charge at pH 7. The fourth group is positively charged amino acids. This class of amino acids includes lysine, arginine, and histidine. Their R groups have significant positive charge at pH 7 and function as weak bases. In nature, there are over 300 different kinds of amino acids. In addition to the 20 amino acids in proteins, some are residues modified after a protein has been synthesized, such as 4-hydroxyproline, a derivative of proline, and 5-hydroxylysine, derived by lysine. Both are found in collagen, a fibrous protein of connective tissues. Others are amino acids present in living organisms but not as constituents of proteins, such as ornithine and citrulline, which are intermediates in the biosynthesis of arginine and in the urea cycle. Cysteine and pyrrolysine are special cases found in just a few proteins. These rare amino acid residues are not created through a post-synthetic modification. Instead, they are introduced during protein synthesis through an unusual adaptation of the genetic code. Some amino acid residues may be modified transiently to alter the protein's function. The addition of phosphoryl, methyl, acetyl, or other groups to particular amino acid residues can increase or decrease a protein's activity. Among them, phosphorylation is the most common modification. These reversible amino acid modifications involve in regulation of protein activity. The alpha amino and carboxyl groups of amino acids, along with the ionizable R groups of some amino acids, are dissociable. When an amino acid is dissolved in water at certain pH, it exists as a dipolar ion or sweeter ion. It can act as either an acid or a base. Therefore, amino acids are amphoteric electrolytes. The degree of dissociation depends on the pH of the medium. This figure shows that at very low pH, the amino acid is fully protonated. As the increasing of pH, the carboxyl or amino group of the amino acid loses its proton. At certain pH, the amino acid presents largely as the dipolar ion, fully ionized but with no net electric charge. The characteristic pH at which the net electric charge is 0 is called the isoelectric point, designated Pi. When an amino acid exists at the pH above its Pi, it will have a net negative charge. When placed in an electric field, this amino acid will move toward the positive electrode. At any pH below its Pi, the amino acid has a net positive charge and will move towards the negative electrode. The farther the pH of an amino acid solution is from its Pi, the greater the net electric charge of the amino acid. The Pi of an amino acid is determined by the negative log of the dissociation constant of the dissociable group. The calculation formula is Pi equals to the average of the pK values on both sides of the dipolar ion. For the amino acids with ionizable R groups, such as acidic or basic amino acids, we should first write the ionization formula and then take the average of the pK value on both sides of the dipolar ion. Okay, let's summarize this section. There are 20 amino acids commonly found in proteins, each of which contains an alpha carboxyl group, an alpha amino group, and distinctive R groups. Except glycine, the alpha carbon atom of all amino acids found in proteins exists in the L stereoisomer. Less common amino acids also occur either as constituents of proteins or as free metabolites. Amino acids can be classified into 5 classes based on the polarity and the charge of their R groups at pH 7. Amino acids vary in their acid-base properties. Okay, that's all. Thanks for your attention. See you next time.